you have 505, and now you have another situation where based on whatever cutoffs you put, like 2-fold change, 3-fold change, or p-value cutoff, whatever cutoff you put, you will get different numbers of genes and different set of genes. Okay. The question is what that data means actually. So suppose if I have a differential gene expression table and I'm just doing a p-value cutoff, I will get 1500 genes. But same time you want also to add a fold change value of 2-fold or more. That means now my numbers will be 600. Somebody else wants some other cutoff, so they will get a different values. But the question is, none of this value really tells us anything about the biology of the system that you're working on. So why it is important? So to do that, you need to understand an idea called as Waddington's landscape. This idea was actually proposed long back, even before genes were discovered, okay, in 1936. And this is called as Waddington's landscape for it's Waddington's epigenetic landscape basically. What is really happening here in this, that you have a cell, okay, think about a cell. When cell is actually rolling through a hill, then each point, like this is a point where cell has a choice to make, like whether it wants to go this side or this side. And one of the example, you are immunologist, one of the classic example is PU1 expression. Then when the cell lineage determination happens, the PU1 expression or non-expression will define which lineage it will go. So similarly, this cell will decide at this point that whether it needs to take this path or this path. And this is every such force, cell will be deciding that, making that decision, and it will keep rolling to the bottom of this landscape. And when that bottom of the landscape will be differentiated, terminally differentiated cells. So since long, like almost till 2005, the idea was that cells once reach its terminal differentiated state, it cannot go back to its original state of or pluripotent state, or where it came back from. But then a classic experiment by Yamanaka proved that no, you can actually revert this Waddington landscape and you can go back into the you can actually go back in this landscape. So that was the biggest discovery. Then what happened? People started actually differentiating. Started converting differentiated cells into its original pluripotent state, and then they do a transdifferentiation. Transdifferentiation essentially means that you started with a neuronal cell, you brought it back into stem cells, and then you actually differentiate into muscle cells. So this is an example of transdifferentiation. You can also transdifferentiate cell from one cell fate to other fate, which is not that easy or trivial, but it can be done. So basic idea here is, what I am trying to actually enforce, that each cell actually possesses a particular cell state, and that cell state is defined by the gene regulatory network underlying this. So if you think about it as a tent, what is holding each point of the tent, those wires that actually you pull for the tent, those tents are actually the gene expressions, the set of genes that are being expressed. So any gene in isolation, there is nothing called as lone wolf or genius or prodigy in case of genes. There is nothing like that. Genes always works in group. They works in networks. And if a gene is changing, if it is not changing anything else downstream, then that expression difference has no meaning. Which means, to know that what is that combined effect of some perturbation, you need to know what kind of functional grouping these genes are falling into. So all the genes in a differential gene expression table will not fall into one or other functional tail, but there will be a group of genes, those will be falling in a group, functional group. And the whole idea of this class is to understand that how you really do that functional analysis and find functional grouping, and what are the different ways you can do that. So let's first do a little bit of theory on this. So one of the common way to analyze gene expression data is through gene ontology. Okay, so gene ontology, or which you are very familiarly known as Go terms, they are structured vocabularies. What is mean? That genes are arranged in various levels, and we will see some of them, and then they are structured through higher level to lower level. Like one of the best example is that genes, all the genes that are present in a biological processes or do some biological stuff. So that is a biological processes. Now it's a broad term, so that is the higher in the hierarchy. And then you have further you go in various levels, it gets more finer and finer. And at the lowest level, you are actually going to find a term which is very specific and will have a very narrow definition. So this is what actually the Go ontology means. So in general, Go ontology consists of three various broad categories. One is called as biological processes, second is molecular function, and third is cellular component. So biological process, as the name implies, it usually deals with pathways and processes that actually defines biology. Okay. The molecular function is basically activity at a molecular level, and one of the example is transcription factors. Best example is that. So if there are certain group of genes that act as a transcription factor, there are certain genes that are kinases. So these are the molecular function, because the kinases function is to phosphorylate. So that means you will be there are ubiquitins are there. So different classes of ubiquitins are there. So that is one kind of a molecular function. Then you have cellular component. So cellular component is the proteins that form the part of the cellular subcellular components, like cells, those are associated, proteins that are associated, or genes that are associated with Golgi body, or something that is associated with mitochondria or chlorophyll. All this, those are actually comes under the cellular component. So basically same set of genes, you are classifying them under various thing, and you are and all the genes need not have all the three functions. Okay, some genes may have only involved in biological processes, some will have only cellular function, but some may have multiple roles also. So this is actually a nice way to interpret the data. So basically it works as a way of parent and child organization. So there is a parent level is there, below that the child will be there, then there is a grandchild will be there. So it goes into that kind of a structure. Okay. So it can be either like it is a relationship between this is A is a part of, or it regulates. So basically it's either it is regulating, or it is part of that process, or it is the process like that. And you can actually do the hierarchical progression of gene function studies using GOTAM. Okay. Now if you look at hierarchy, I know that this is fonts are small, but here you see there is a biological process, and then it's involved in regulation, then regulation of biological process or cell process. And so basically what you are doing, you are actually structuring a bigger biological process into its parent-child relationship, and finally you are coming to this term which is very specific, which is negative regulation of, say, programmed cell death. So at that stage, when you are actually crossed all those hierarchies and came at the lowest level of the data set, that means now that term is very specific. This is only related to negative regulation. So even not regulation of programmed cell death, it is only negative regulation of programmed cell. So you can go up to that finer level. Most of the analysis what we are going to see today, we are allowing program to choose its own level. So program will actually go through all the levels and find out a level which is most suitable for it. But then you can also enforce levels. Levels are between 1 to 7, and you can say level is equal to 3, then it will actually take the third level of that hierarchy and do the enrichment analysis for you for that. So you can also enforce levels, but in generally the programs automatically allows you to choose between the levels. Okay. Now the goal of a statistical goal of a GOTAM analysis is to identify Go terms or gene ontology terms that are significantly enriched in your data set. And then how you do it, you do Fisher's test or chi-square test, and depends upon what kind of data sets you are using. Most often it is the Fisher test or hypogeometric test is being used, and then you do a multiple testing correction which is basically either Benjamin-Hochberg or Bonferroni correction. I actually tried to explain this multiple to some people yesterday. The idea here is that if you are asking same question 1,000 times, there is a chance that you are going to get a false positive answer. So in this kind of analysis, every pathway you are taking or every ontology term you are taking and are asking, are you differentially enriched between my sample and the universe or not? If that is the question you are asking. When you ask this question repeatedly, there is a chance that you will get a false positive response, saying like somebody will randomly say, yes, I am. So to do that, you need to correct. One of the best example which I was talking to some of the people yesterday, the way I gave that they told me that Manpreet is the name which is most common name in Chandigarh. So if that is the case, Manpreet is the most popular name in Chandigarh, and I want to meet Manpreet, and I don't know who is Manpreet, and I only know that okay, there's a person called as Manpreet, and she is from PGI. Now my choice is like, if I came to Chandigarh, and every single person I meet, so I just know she's a girl, or maybe Manpreet can be a boy also, no? Yeah, it's a gender neutral name, so that makes it more complicated. So every person in Chandigarh, I will go one after another and ask, are you Manpreet? Are you Manpreet? Randomly there will be a person who will say, yes, I am Manpreet, because that person is Manpreet. This is the most popular name in Chandigarh, so it may be boy or girl. At that point, if it is a boy who is actually responding, or man is responding, then I know that okay, you know, it's possible that it's a false negative actually. But at that, and then if suppose happen to be a girl, then I need to actually also add a second criteria, which is, oh, then I will ask, are you from PGI? Then she say, okay, if not, I am not from PGI. If actually that person is also happen to be for PGI, then I need to have another criteria to figure out the Manpreet I want to meet. So this way, when we are actually doing same test, when we are doing thousands of times we are asking same question, then there is a chance, and random chance, that you will get a false positive, and you need to correct it for that false positive. And there are two different ways to do that statistically. One is called as Benjamin-Hochberg correction, and second is called as Bonferroni correction. Unfortunately, both of these corrections are harsh on the data sets. Okay, so that is why what happened. There are chances like you may remove some of the true positive also in this correction process, and but this is actually a very conservative way of doing it. What people found that there is an alternate way to do such a corrections, and which is called as FDR, or false discovery rate. In case of false discovery rate, what you are doing, you are first measuring the distribution of p-value, and based on that you are creating a statistics for false discovery rate, and that will give you a little less conservative way of estimating the correction, and then that's how you can do a multiple hypothesis testing. So in case of bottom analysis, both ways, FDR as well as Bonferroni and Benjamin-Hochberg, both ways actually people are doing. Some places you may say a value called as Q value. Q value is actually related to FDR. So some people say that as FDR, some people say Q value, but if you are asking this to a statistician, they will say I am wrong. So but in fact, you can actually use for a practical purposes, you can use those two terms interchangeably. Okay. Now, how to do enrichment analysis for GoTerm? The idea is very simple. There is something called as universe. Okay, so GoTerm is a curated database. So somebody is really reading a paper manually and curate that database. And when I was in Jackson lab, the person who used to sit next to me was doing this. His job was like whole day he will sit in a room, and his only job is to read paper, and based on that he will curate that database. So you think about how boring that job is, but he was happy a lot. He was doing that. So anyway, so you have a universe. So all the genes are not there in Go database yet, and that is one of the major limitations of such databases. So you have only 6,000 some genes are there. When last time I checked, 6,800 genes something. So that is the universe. Okay. Now you have a differential expression table that you, whatever criteria you actually had, so based on that you created a differential expression table. Now that gene has some number of genes. Now what you are asking, that you take one pathway, say suppose you are taking wind signaling as a pathway. You will ask how many genes are there related to wind signaling in my universe. So which is this X. Okay. That means you will actually subtract X to M. M is actually the total number of genes that is there in the universe. So you subtract M minus X, and you will get this Y value, which is the number of genes in my universe which is not wind. Same thing you do with your differential data set. You will ask how many genes are there in my differential gene expression table which is related to wind, and so then you minus that with differential expressed gene, then you will get Q value which is not related to wind. And now you actually do a F test, because this is a 2 by 2 contingency table, and you just do a F test and you will get a P value, and that will tell you that whether this is significant or not. And when you are calculating fold change, you are basically calculating x divided by y and then p divided by y, and that ratio if you take, that is the fold change value for your Go term. So, this is the process that being done, and this is done for every single term that is there in the Go database or KEGG pathway database, any database, whatever number of terms are there, the program will do that in the backend for each term, and they will calculate the p value, they will calculate the, and then they will do a correction for the p value, because now you are doing this test hundreds of times, different pathways that need a correction, and once they are corrected, they will report the based on p adjusted value, which is significant. Okay. Now, so in generally what happened, the input list for most of the analysis which uses over-representation analysis, the input genes are ORA, or input lists are significance genes. So, you will make a criteria for significance and you will put them, and then you will compare these frequencies, and then you report based on adjusted p value. So, there are many such tools are there. Manpreet is going to talk about David, but meanwhile I will be talking about cluster profiler, which based on R, and then there are Python based other tools are also there. In case of gene enrichment analysis, what happens, one of the key question is why we should have a criteria at all, like two-fold change. What is the sanctity of such a fold change biologically? I mean, do really cells care, like if a gene is expressed 2 times more or 3 times more or 5 times more? No, they don't, really they don't. But this two-fold change idea came from cancer biology. So, all the microarray research started with cancer biology. Okay. So, when cancer biology data were being produced, they have large number of genes which are getting changed in such experiments. So, if they set up a two-fold change criteria, still they have enough number of genes to further analyze downstream, and then get some kind of a biological insight into that. So, that is how this two-fold arbitrary cutoff came. But that stayed as if like everybody you ask, they have no idea why they are using two-fold, but everybody is using two-fold cutoff. So, there is nothing like such a cutoff. Biological system works with a small incremental effect, and if you ever actually work with deletions like loss of function mutants in mouse and others, you will see the changes, that gene expression changes are very small, not very big. Very few genes actually changes huge, but most of the time the functional genes changes make small changes. So, in case of this kind of gene set enrichment analysis, rather than doing a cutoff based on log two-fold change or something like that, just you take all the significant genes based on p value, and then you go and put all those genes together in the analysis. Idea is that even the smallest change in a gene expression, but it is a part of a larger set of a network, that means that change is significant. So, now you are not relying your observation based on the magnitude of change, but even the smallest change can be considered as a biologically significant. So,
what should we take? P value or adjusted value?
Adjusted, always adjusted p value. P value, especially with any kind of this kind of data, p value, unless it is not corrected for my multiple hypothesis testing, those p value has no meaning. Okay. So, basically this kind of analysis will avoid any kind of arbitrary threshold, and it also actually includes some kind of a permutation testing, and we will try to see GSE if we could run them from this time. So, one of the things that you always think about when you are doing this kind of analysis, you should always stick to one kind of data sets. You can do analysis using either various kind of IDs like ensemble ID, gene symbol, and enterize IDs, everything. You can just use any kind of ID, but whenever you are doing analysis, stick to one kind of ID, and then every time use that ID for analysis. You can also create a background set, which is a good practice. You can always do that. Look at actually always use the updated Go term analysis data set. One of the challenges is that if you are doing a Go term analysis like now, and if you do after a one year or so, the new terms might be added, new genes might be there. So, which means that every time you do this analysis, not like in a day or two, but then if it is done 6 months or 8 months after, you tend to see a difference in your results. So, you should always be aware of that, and so that it's always a good practice to redo even the old analysis and make sure like when you are comparing your data, then it's all done together. Okay. The other thing is like most of this analysis, they will give you some random pathways also. And the reason why that happens because same genes might be involved in some other pathways, and I will actually come back to that sometime later, but if I find time I will tell you why. So you have to also think about the biological question that you are interested in. If you are working on a heart development thing and you are finding a viral pathway in that, you know that that is not real biological. So you can definitely omit that based on your domain knowledge. And so basically at some point when you have that pathway in your hand, it's always important rather than just reporting what is computer is giving, rather than think about it, see like if it makes sense to you, or what are those pathways in your significance list gel to your question that you are asking, and can you test those hypotheses? Okay. So one of the major problems a lot of time happens is the outdated Go term. We already spoke about it, and then ignoring multiple corrections. So you have to always use adjusted p-value, not the p-value. Then we also look at that how to avoid this issue, over interpreting generic or redundant terms as we go. And then also it's important to validate your finding from an independent way. Like suppose you get a cardiomyopathy related pathway as enriched one in a mouse experiment, you need to check for that function, like is my mice is actually getting those phenotypes or not. So okay, is the pathways that you are getting, okay. Then finally we are actually this is basically what I was talking about. It's a structured data sets, statistical enrichment is done, and then you need to combine pathways and other things with. One of the other pathways which very popular among many of this is KEK pathway, which is basically for signaling pathway. The good thing about KEK pathway is that it's a hierarchical in nature, and it shows you what is the impact of each of this gene over a downstream gene. So this is a KEK pathway, and one of the major bottleneck for many of this analysis is that there is no such hierarchical relationship exists in this analysis, because you remember like how we are doing this analysis, we are actually asking if this is present or absent. Here actually we don't have a weightage. So I can actually quote you two people. One actually Allama Iqbal, he has a very famous Urdu share कि जमहूरियत वो हुकूमत है जहाँ बंदों को गिना करते हैं तौला नहीं करते. So basically what he was saying, that in a democracy you weigh, you count people, you don't weigh. And exactly that is what we are doing in Go term. We are not actually weighing the impact of those genes, rather than we are just counting them. So the impact of this gene, which is far upstream like TNF gene, is actually way more pronounced than a gene in the periphery of this network. So if you have a perturbation in this gene like TNF or trail, you will have a bigger impact over the downstream cascade network. So, but none of this analysis, enrichment analysis or aura, actually take into that account. And if possible, I would like to show you this one. This is called as PIA, or signaling pathway impact analysis, where what you are able to do, if two genes, say this is a network of gene, then if you have these two genes have changed its expression in your differential gene expression, which means they are just lone wolf. There are random genes that are actually changing. But suppose if A is changed and B also changed, then that means you know that the impact of change of A is actually passing through B. So you can actually predict the perturbation effect of this. So any of these two genes perturbed, you know that what is going to happen to the further downstream pathway, and you can make a precise calculation for the perturbation and create a p-value. And when you actually create a pathway and enrichment analysis based on such, when you are including the perturbation and weight of the gene in your analysis, and which SPIA does, you will be able to incorporate that also. And then finally what you also can do, you can predict that what is going to happen with the pathway. So one thing is about the gene, that whether gene is going up or down, but you can also say end result of this perturbation will be that, like say Wnt signaling, if I get Wnt signaling is inhibited. So that inhibition or activation part you will get. In that case, once you have that kind of a interpretation, then you can go back and design your next experiment where if suppose Wnt pathways are inhibited, then use a Wnt activator and show that you can rescue the phenotype. So that means you have a functional validation now based on that kind of. Okay. So I think this is the presentation I want to give, and let's start with. We did not send that. Francis, can they access those notebook there also? No. Oh. Okay. You open the 4th day.
So, when we select genes by the p-value, right? We select p-value 0.0. That's quite common. What it means? See, we select some independent genes, but the 0.05, that 5%.
The chances of selecting it
Is the chance of type 1 error. What is the type 1 error? Means the probability of having a false discovery. So you're saying if you do that and you have 20,000 genes, 5% of 20,000 people to the 1,000 genes are false. You're going to have a significant p-value, right? They're going to be false discoveries. So I think it's good enough reason to assume that you're going to have to correct. If not, you have 1,000 genes that you're going to be selecting that are going to be false discoveries.
Okay. So.
Sorry, did you see the new file? I put a new file in the folder. Let me know if you don't see it.
the name of the file?
Functional something. Functional analysis is showing up.
Yeah.
Not opening.
They have that combined D data set also there?
No, okay. Give me a second.
By that time Francis is putting there, I will just show you Cold Spring Harbor. So this is CSHL. So this is called as Cold Spring Harbor Laboratories. It's in Long Island. And if you go here, I believe yeah. If you go to events, there is something called as meeting and courses. And it's a lot of meetings and courses are there. If you go to courses, I'm just showing you all courses. So see. See here you have tutorials in genomics and bioinformatics, RNA-seq analysis, which is from November 9 to November 11. So I think you are a little late for this course. Advanced sequencing technology and bioinformatics analysis, again that is same time. Scientific writing retreat, computational genomics, which is in December, methods in cancer neuroscience, cryo-electron microscopy, quantitative imaging, cell and developmental biology of Xenopus. Like that, you can see like large number of courses, very specific, like ion channels in synaptic and neuronal circuits, a very specific course. Mouse development, which is a very general course. Then this is the one which we go, I think I went for statistical analysis for genome scale data. They used to call us statistical analysis of functional genomics, the pancreatic cancer, then brain tumors, advanced technology in molecular neurosciences, Drosophila neurobiology. So very interesting advanced topics. They have 2 to 3 weeks. Some of them are 1 week courses also, but most of them are 3 weeks. And you can find that here. And then if you go to meetings, and this is conducted every year, not like 1 year. Then you have like all this kind of meetings are there for them. Like single cell analysis meeting is there. Then you have genome informatics, zebrafish neurobiology, plant genome system biology engineering. So these courses and meetings are very good, and they also give you students, especially they give travel grants, and DBT also have something called as Women in Science, and they actually give travel grants through Women in Science, so you can actually get that through DBT also.
Like for the proteomics and all, we use usually daily time for the data analysis. But
Yeah, you can do same thing here.
same.
Basically, once you have the, it's only using the gene IDs. Okay, so
So for this SPIA, what is there any other software where we can get those
no,
information?
this is SPR needs to be done on bioconductor only, R only. I think the Ingenuity software from Quadrant, I think that uses SPR as a backend. Um, uh, yeah, they are paid, but that is what actually they
What
ask me.
is the difference between what we do that the string pathways and the cytoscape?
I don't
And
know. I don't really believe any of those things, strings and cytoscape. I don't know why. What the?
Way of, making your data look jazzy.
Yeah, I
This
mean,
kind, that
none of
kind.
those makes any sense. Ever they made any sense to me, unfortunately, to be honest with you.
so they also do show some nodes and the highest specificity and
And I don't know. I mean, I have my own problems. Probably wrong. Probably I'm a priest in a wrong church.
no, it's okay. We are learning here.
No. It's not about you. It's about actually, you are right, like a lot of people are using it. So it doesn't matter whether I believe or not. Okay. It's simply like
But it
even,
should make sense when we will
yeah,
see
but it
that.
never made sense to me because I never understood that what part of the biology they are trying to. Yes.
Didn't make sense to me. It makes sense to me.
No, but my problem was mostly like, I could not figure out that how biologically that is even possible. Like you have differentially expressed gene, and then now you are saying like they are part of a network. So those two things are very different things. Okay, network of because I'm a biochemist also by training, so the network of a gene networks, like when it is interacting in a network, it usually there's only two kinds of relationship. That one gene regulating other, and other region regulating other. Those things you can capture through most of this kind of analysis via algorithm or anything like that. But then what they're talking about is like if it is a part of a larger like cytoscape and all this, it's complicated. I don't know. I always thought that there is some biology missing there, and I could not spin
So
my
how
head.
come one is involved in so many times that it's the same?
It is actually what say. So that you have to think. So that you have to think from evolution. Okay. So
It's ready, Bob.
it's ready. So gene regulatory networks, basic. Okay, guys, I have a question for you. Okay, a bit of a philosophical question, but I want to know that what do you guys think about it. So one of the argument for Darwinian evolution is that the genes are conserved. So some or other form, even you look from single cellular organism to multicellular organism, the same genes are actually conserved. So tell me one thing, why do you think like genes should be conserved? What is the compulsion of genes to be conserved? I mean, there are 20,000 genes. They always can, lose or gain or make a new gene and can do same thing. But genes are conserved. And why they are conserved? Why you think there is a inherent compulsion in evolution for genes to be conserved?
Otherwise, there will be so much change over the generations.
But that is good, no? Because whole evolution is driven by variability. So more and more variability you induce in a system, there is a chance that you will evolve into something bigger or better.
Something drastic.
Even drastic, what is the problem with that? Tomorrow it's zebra, like
Whether your system will be able to handle that.
So that evolution is
no,
also
even directive, it doesn't matter that whether it's a directive or not. But the important thing is like if a system can create variability at a faster rate, like in case of viruses and all, it's advantageous.
Your system will
Or
not
the
be able to
bacteria.
handle that.
Eh?
Your system will not be able to handle that. I got deleted from
no,
the
we are
system.
not fighting for survival.
Who told you? We are not now because we We built house, we built medical system, and we built PGI, so we are not fighting for survival. But still the animal in the forest, they are still fighting for survival every single day. A tree standing in the rain, it is surviving, fighting for survival every day. It's fighting against insect, because tree cannot spray insecticide themselves.
Is it because even from the most primitive organism to the most evolved ones, so-called evolved ones, the basic mechanism of gene, I mean, transcription is almost similar, isn't it?
So yeah, I mean that is the idea, but I will actually put it little more refined way. I understood what you are trying to say, and that is what the idea is. Anybody would like to attempt one more time?
But like in case of COVID, because we have generated vaccine, so another strain comes, so that came fast. So that means that virus was surviving for their survival. Fighting that.
Yeah, but that is advantageous. No, that is advantageous. No, no, but in normal atmosphere also, when you don't have vaccine, virus actually mutate fast, very fast. They actually evolve. The new strains comes. And bacteria does that all the time. Many organism does that all the time. So there is nothing wrong being fast, okay, unless you are not driving in Chandigarh roads, you can be fast, okay, still fast. But the issue, it's not about the fast. So what is really happening there, that the idea,
There's no time for some experimentation happening that will
no. So the explanation is more simpler, okay. This explanation is way more, and actually that is, oh.
So during cell division, the chromosomes they are derived from the previous cells, so they are conserved in that way that they will not change drastically because one set of chromosome comes from.
No, but that is also not true. See, you think about cancer, how drastic changes has happened in the cell, and everything changes, like the cells don't look even anything like what the tissue of origin is.
That drastic change, that is not surviving.
They are surviving. Cancers are better survivor than, no, the cancers are better survival than the human being who is carrying those cancers. They actually relapse, they metastasize, everything they do, they play havoc on the system, and even body's own immune system cannot do anything about it.
The basic mechanism of life is so worked out in the primitive to the genetic. I don't want to mess up.
No, why that is worked out? That's the question.
If we genes aren't retained, then they basically become more mutable, right? And at that point, you lose out on, basically you don't have the chance to prove that this trait will survive or is.
No, okay. Let me give you a clue. Okay, so whatever be the answer of this question is, it has to do with this class. If actually you put your answer in perspective of what I am going to teach or what I was teaching for last 40 minutes.
It will not be in harmony.
So the issue is that gene actually works in networks, and basic gene regulatory networks are conserved across species. So the same module of genes or set of genes are actually used again and again and again during evolution. Yeah, it's not that why do you want to change. The problem is that when so it is like you are 5 people are actually going for a hiking, and each of you are given a role. It's very hard for one person to deviate from that. Otherwise, you are very arrogant. You are full of yourself. Unless you are such a person, it's very unlikely that throughout the course of your hiking, you will deviate from that path, because a role is assigned to you, and it is not because it is because of that group dynamic. Group is actually has to behave according to the roles assigned, and that means you will less chance that you will veer. So that's why genes don't mutate in a evolutionary time, and that is the first thing. Second thing, the same pathways are reused in like heart development. If you look at vertebrate heart development, the same basic central network is actually, the FOXP network is actually used again and again and again. Similarly, if you look at body forms like bilateral symmetry generation or neuronal symmetry like anterior posterior axis generation in any organism, bilateral organism, it's always Hox genes, and how they pattern Hox genes based on anterior posterior polarity. So the same network, same set of genes are reused again and again and again across the, and this is only possible because genes works in networks. They don't work alone. and lone wolf has no role in biology. Okay. Okay. So let's start. You have a Jupyter notebook. This is called as functional analysis, and you have a file which is this one. You can just open and see what it has. I just actually made a small file for you. It has gene ID. So this has gene ID. It has base mean value. I'm not going to use it, but just it is there. Log 2 fold change, which I'm going to use. P value, P adjusted value, and then I think it has other 2 columns, which is two more IDs. One is entrace ID, another is ensemble ID. So I have this. Okay. So I have 3 kinds of ID in this table. I have log 2 fold change. I have P value, adjusted value, and so this is a table. This is the classic table that you get from the differential gene expression data. And unless if you don't have any of these gene values, like any kind of symbols, entrace value or ensemble values, I will actually, if I have time, I will show you how to get those values. Otherwise, there are other easier way to do it. But let's believe that you have all these things. Okay. This is a notebook. Essentially, you don't have to load this library, but for some reason, I am actually not able to get it automatically loaded. I don't know why. So what you do, just go to this column, cell, and run that cell, like as you do, shift enter, and it will load you all the packages that we need. Okay. Just do a shift enter, or you do run, and you will be able to do it. So it just loaded all your packages, and we need few packages like cluster profiler. We need path view. We need SPIA. We are using human samples. So this database is HS. If it is mouse you are using, then you use M capital and small m, or if you are using Drosophila, use big D and small m. Okay, and then other databases are there, but these are the major common organism that people use. Now, so this is running, all thing done. So now we go to our file. So this is familiar to you. We have used it many times. So these are a CSV file, and we are going to read this CSV file into an object called as gene list. So we will do that, and so we did that. I'm going to do something. I'm just going to add one cell here, and I want you to do so and just check this file. Okay. So how you will check? You will do a head, and you will say gene list. Okay. And I will see, okay, the data looks like this to me. This is the first 5 lines of this data. There are some gene names are there, and all this different columns that I need is there. I can also check what is the dimension of this file. So I will use dim, and if I do a gene list, then so it has 25,665 genes or rows, which is actually each gene is probably for a gene, and then you have 7 columns. Okay, so just we are exploring our data set. Now, next thing we need to do. So next thing we need to do is to subset this data set. So as I mentioned, like all this Aura kind of like enrichment analysis platform, the biggest challenge is that we need to take responsibility. So as Spider-Man said, with great power comes great responsibility. So we also have a great responsibility, and one of the great responsibility we have is to subset and find the right criteria for subsetting, and there is no criteria such as right criteria. I am just using a criteria here. I am just saying if the absolute log 2 fold change value is 2-fold, that means 2-fold up regulated or 2-fold down regulated, then yeah, okay.
Click on the folder and close that section completely.
And Okay. So I'm using a command called as subset. This is a base R command. You can actually use what Francis told you in the dplyr also, but then here actually I'm using a subset command, and what I want to subset, I want to subset my object called as gene list. So this is there, and based on which column, I am based on the log 2 fold change column, but I'm just want absolute value. The reason why, some will be positive direction, some will be negative direction because of up regulation and down regulation. I don't want to actually distinguish between them. So anything that is going either twofold up or down, just subset them and give me that, and store that in a new object called as gene. Okay, so I am saving that into a new object called as gene. So now I have two objects. So are you are able to run this? So if you look at dimensions of gene object, you will see there are 509 genes. So basically I have 509 genes which is twofold upregulated or downregulated in my list. I'm just kind of not using p-value here, but then you can also have multiple criteria, p-value and others. Okay, now let's actually, and in this notebook I have added some notes also for you to read, that what it does, what this terminologies means and everything, so you can always read them. Okay, now we want to do a Go term analysis. So as I mentioned to you, that there can be three different ways of doing Go term analysis, or three different hierarchies are there. One is biological processes, molecular function and cellular component. So the first thing what I am going to do, I am going to do molecular biological process, and I want you all to do molecular function and or one of them, CC or MF, whichever you want to choose, you do that. Okay. So what are the terminologies, that what are the information that we need? We need here, this is the function, this is a function from cluster profiler. Okay, enrich Go, this comes from cluster profiler. We have subsetted our object and we saved them as a gene new object which has 509 differentially expressed gene, and then we want to take the gene list from ensemble column. So that is why we have a $sign and ensemble. You can also use with the other IDs also, but then I am actually using this enrich Go, takes usually ensemble. Then you have to give what kind of organism you are working with. So in my case I am working with HS or human. If it is mouse, then that will be org.mm eg.db. So you will use organism database as the organism you are using. Now third one is what kind of key I am using. What is the key means here? Here actually the what kind of gene IDs I am using. In case of me, I am using ensemble ID. So I said like my ID is ensemble. If I am using symbol, then it will come, symbol will come. Ontology, what ontology term I want to, in this case I am doing BP, yeah, or biological process, but if you are doing molecular function or cellular component, you will use MF or CC, and both in capital. Okay, no small letters there. Then what kind of adjustment I want to do? It's actually Benjamin-Hochberg correction, that is why BH is mentioned there. Okay, and then there is a p-value cutoff and q-value cutoff, and forget about this part. Only thing you should remember, that when you run this command, whatever organism you are using there, you should use the same organism database here. Okay, and then if you run this command, it will take few minutes to complete. And it's doing all those Fisher's test and adjustment of p-value adjustments and everything. And if you want to run, you can also run molecular function and CC along with me, make the changes and then just run them. Would you run?
So what is this Q value?
So Q Q value is actually equivalent to FDRs or it's adjusted p value basically. Either way you can, people uses it. That is not the real, I mean if you ask a statistics person probably they will get offended, but for biologists Q value can be FDR, adjusted p value equivalent to that. So for us it is fine.
So you told that we don't have any correction or those correction are harsh and we may lose out on two positives. FDR we are using. So why have we kept that?
Most of the statistics actually based on FDR is a relatively new way of doing it. So many packages are actually still using all BS1 or Benjamin-Hochberg corrections. Okay. So my run is over. Okay. Now I actually want to plot them. And so before that what we'll do, I'm done with this. So okay, here if I go here in the dot plot section and I will add a cell, okay, and then I will ask head and I will say ego 2 because this is the object that we created. You see this one top, we have created that object as an output for my ego 2. That was my output for my Go term analysis. So I want to see that if I found any terms in that or what are those terms. Okay, so I just want to see a head of this and it will give you, and don't worry about much of these things. So it actually tells you a few things here, that what are the Go term ID and gene ratio, which is basically fold change in this case. Then you have p adjusted value, q value, and this gives you a list of genes which are these are actually genes here, but these are RSV data. But forget about the biology of it at this point. We are just looking at how they. So it's a big table and you can write it out, like you can write as an Excel sheet and you can use that for, like you can handle that as an Excel sheet also. Okay, but we are not actually interested in that. What we are interested in, can we actually make some nice plot out of this? And one of the beautiful plots we can make is a dot plot, and that is pretty simple. See, with just single command dot plot and within the parenthesis ego 2. Ego 2 is the object that we created from our Go term analysis, and it gave you a beautiful dot plot with all the enriched terms here. Then p value is actually colored based on the based on this matrix. Like lower the p value it is more red, higher the p value it is actually blue. And then the size of these dots are actually according to the count, how many genes are there represented in your data. That is how, if more genes are there, the dots will be bigger.
On the x-axis, it is written as gene ratio.
It's a fold enrichment basically. Gene ratio is essentially the fold enrichment analysis in this. Then same thing you can actually also plot as a bar plot. Okay. So say it's the same data. Now actually you are being shown as a bar plot. So instead of dot plot, I like personally dot plots more because it looks a little more cleaner. But bar plots, a lot of people like to actually put them as a bar plot too. So this is a bar plot. Again, in this bar plot, they are on the x-axis. They are putting count. So basically, this is the number of genes in your differentially expressed table with that terminology. Okay. Then this is another way of making bar plot. It just showing only the 12 categories, but you can restrict anything. But the main thing I want to actually, these are like simple plots, dot plots, and those kind of thing. But I want to actually show you two plots, and that is the key plots in the analysis of gene regulatory networks or this kind of analysis. So one is called as EMAP plot. Okay, and you have seen these plots actually in a different context, but this one is called as EMAP plot. What I will do, I will actually plot them as PDF. I think that is better, and I will save them as PDF because there is a why I will show you why I will do that. So let me actually explain you this command. So you have seen Francis has used this many times. So PDF, and then you want to save this in this name called as EMA ego2.pdf. So this is the name I want to save, and I am putting a width and height argument. And the reason why, these are big pictures, and if I actually use a default which is 800 by 800, then I won't be able to see it. So I am just doing that. So and then this is a command I want to use, emap plot ego for the ego2, and then this I'm actually closing that plotting engine with the def.off option. So just let's run this, and I believe I have the plot. Where is the plot? Yeah. So okay. Oh, this is okay. Let me actually do on something else. This is it's saying like dimension is too big. I will go for a PNG and then PNG. Okay, so this is your EMAP plot. Okay, and do you know what we are seeing here? No, not networking. What are we seeing here? Nodes of what? So no, so in this actually they are not genes. Okay, so what we are seeing is that how, suppose you have 10 or 15 pathways got enriched.
Some genes, how they interact with each node is a pathway or a
Not genes. Here it's not talking about genes. So EMAP, what EMAP plot shows you that you have 10 or 15 pathways that you saw the enrichment. Now the question you are asking, are these pathways are related to each other? Basically, like you are looking for a bigger, what is their relationship with each other? In this, all the pathways are related to each other, but there will each red is a pathway. But you know, there are instances when all of them will be separate. Like you will be having like 3 or 4 pathway together, 2 will be there together, 1 will be like lying out. So you know that okay, there is a module. The modules of pathways are also, and they are how they are interacting with each other. But I will actually go to next plot. Then we will actually come back to this again.
The dimension for this? Change dimension.
It's 200 by 200, 2000 by 2000, and because it's a bigger plot, that's why I wanted to.
2020.
Okay. Sorry, I'm actually going a little fast, but my okay. Now the next plot that we are working on, the next plot that we are plotting on this same data is called as CNET plot. Okay, and CNET plot is what actually Dr. Amit was just mentioning. It actually shows you the relationship between genes. So you got all these pathways. Okay, now you see. So this is if you look at, it is showing you that how genes are actually related to this pathway, and if a particular gene is shared between multiple pathways or not. Okay, so this is what actually you can do this kind of analysis, this kind of plots you can plot and do the analysis for your. Now,
Can you show the command for this? CNET?
yeah, it's just same.
But just to run.
Yeah, sorry. हाँ वो हाँ जस्ट उसको खाली रन कर दो नीचे आ जाएगा वो ओके anyway so for most of this kind of analysis you need this four kind of plots you need to have a dot plot or a bar plot and then you also need a CNET plot and EMA plot to see how genes are related to each other or how and I will actually show you one other example because I was actually doing one analysis here this is my because that has little something different okay here yeah so let can you see this so this is a different data set okay I thought I should have used it so now you see here you have this pathway here you have a second pathway you have a third pathway now between these two pathways these genes are shared these genes are unique for this particular pathway. Similarly, these genes are unique for this pathway, but these genes are shared between these two pathways. So, the enriched pathways what you got and the genes underlying those pathways, how they are shared, you are able to figure out, you are able to find that using your CNET plot analysis. So, this is a different data set I actually I had. And similarly, I am going to actually show you a EMA plot also, EMAP plot also on the same data set. And then you will realize that what I am trying to, okay, I don't know whether. MAC or PNG. Yeah. So here is a EMAP plot, okay, for that data set. Now you see, these all of these pathways are actually interrelated with each other. Okay, this is one set of pathway, they have some kind of a relationship. Now you have another set of pathway, they are interrelated with each other. You have a third interrelated pathway, but while there are 3 or 4 different pathways that are just, you know, in most of the cases, these kind of pathways are, even if they are enriched, they may not have much of a significance. Because if these kind of pathways, there are possibility that there are multiple pathways coming as an enriched pathway, but they are part of a module, then likely that they might be more significant or the one something that you need to pursue in your experimental data. So EMAP and CNET plot, you saw like both of those plots are very useful. And why you do EMAP, CNET plot, one thing is looking at the interaction, but what are the other reason why you do use it?
Just picking up the one particular gene where the
Yeah, basically the thing is, sometimes what happens, only one set of genes will driving everything. You have 10 pathways, but all the 10 pathways have the same gene. That means only one of those pathways are right, not anything else. So CNET plot will also give you what kind of diversity is there in the gene usage when they are doing this kind of enrichment analysis. Okay, so is this useful? Say something. Yes? Okay. Now, you are looking at Go term. Many of you might have done molecular function or or CC. So basically, you can just by changing one small command ontology, you can identify all the 3, and whatever output you are setting, you can actually run them through all the 4 plots, and you can generate your plot.
which software?
Oh, it's R. It's R actually, you are in hardcore R, you are doing coding. Okay, and you can actually, so what I was using just now I showed you, that was R Studio. So you can run all this packed thing in the R Studio, and I will send you a RMD file for that too. I will tell you what to do. Okay. Now, KEGG pathway. So similarly like Go term, you can do a KEGG pathway analysis. Okay, and then so this is the code that you will use. Here everything is like clear. You are using entrust IDs for the genes. So same, the object gene you are using, organism what you are using. So in the KEGG pathway analysis, it uses terminologies HSA, MMU for mouse, HSA for human. So we are using HSA, and then we just gave a p-value cutoff which is 0.05. And once we are done with this, I think it is done. Yeah, it is done. And if I want to see if I have any term, then I say head and KK. That is actually our object, and it shows you that various pathways that are a Go term. Now this is giving you the KEGG ID, KEGG pathway IDs, and then its enrichment values or p values are in. Okay, and then if you take that KK object, and you can make dot plot, you can make bar plots, you can make EMAP plot, you can make CNET plot, and you can depict all of them. Okay, and then we will come back to this when we are going to do a path view analysis. Okay. Okay. Now, so I am not going to do the plotting. Now, you remember like I was talking to you about the problem with Go term analysis. We have to take larger responsibility of setting up a threshold. But many of us, I think we should not take that responsibility unnecessary. So GSE analysis actually uses, actually take that burden off from us. So if you have a differential expression gene list, you don't have to make any cutoff. Rather, you give whole gene list, and then you tell program to actually analyze the data based on, even small changes will be taken into account if it is a part of a network. And that is what we are doing here. The first part of this analysis, what you are doing essentially is to just clean up your data. You are just cleaning up your data. So you are taking the gene list, which is the all genes list, and then you are taking an entrust ID, and you are removing any row which is NA. If the entrust ID in a row is NA, which is not there, then you want to remove that row from your analysis. Okay. The second line, you are actually removing any duplicate row. If you have a row where the entrust ID is duplicated, like both the rows have same entrust ID, it will actually remove the second row. The first row it will keep, and it will remove the second row. So you are first line of this code, you are removing NA. The second line of code, you are removing the duplicate, and then you are passing off the log2 fold change value into a new list. Just it one log2 fold change value, and it is leaving that to a list called as GSEA list, and you are adding the name of each of this one from the entrust ID. So this is, you actually created a list called as GSEA list, which has no duplicate entrust ID. It has no NA in the entrust ID column, and you have a clean. NA usually what happen, no, there won't be any gene name. So what happen, programs usually put NA there. So they don't want any row which has, no, there is no ID. That is why NA is there.
But that number that
the symbol might be there, and sample ID may be there.
symbol is not there, but that ID is there.
Which ID?
So that ID gene has not been identified. But I mean,
Maybe, yeah, maybe. No, other way around also. Suppose you have a gene symbol. Say I discovered a gene called in my name, like Boni De Kumar. I gave a gene ID like gene symbol as Boni, BONY, and so ensemble also I got, but entrance there is no ID. So that means when I am actually annotating that gene for entrance ID, it will say NA.
I'm saying that entrance ID is giving a number.
Yeah, it will give a number, but instead of that number, there will be NA. If that number is not assigned to that gene.
Three ways of depicting a gene.
Yeah.
Ensemble, entrance, or that one.
Yeah.
Gene ID. So we are picking up only this. If
This
there
one.
is NA,
Then this NA we want to remove it, and if there is a duplicate also, then we also want to remove it. Okay. So just run this line of code, and you are able to generate a gene list. Okay. We created the gene list, and Then what we are doing, we are actually doing same thing what we did with go term. So only difference is that the gene list what we created up, we are taking that and running into and a new function we are using called as GSE go, and then we are creating and then we are using this object, we are using organism, and same like we are either we can use biological processes, molecular function or cellular component, whatever you want, and then just run this. Okay, it actually also do some permutation, so that is why it is asking for n perm, how many number of permutation you need to do, so do all those kind of stuff.
I forgot इसमें और पहले वाले में क्या था?
बस पहले वाले में हम कट ऑफ करके उसको लिस्ट देते थे,
अच्छा।
और यहाँ जो है वो हम कट ऑफ सारे ले लेगा, उसके हिसाब से वो करेगा वो, ठीक है. सो लेट्स रन दिस एंड या सो same like go term, and then we are going to make all the plots, whatever go term plots we made. Will you guys will be able to do it yourself? No, I'm saying like after this. See, the thing is, you can run same set of code. Okay, only thing what you need to do is to change the first first object and create the object. And if your header names, like the column names are different, just change the column names and just run it, and it will create output for you. Okay, so you don't have to really rewrite all of this code for your rest of the analysis. So I'm going to see if I got anything. So head ego 3. So see, I got some terminologies. I got some terminologies here with the IDs and enrichment values and those kind of stuff. So you can do this for biological processes. You can do this for but you will get a different terminologies compared to. Okay. Now. Okay. Now the question is, I want to show everything together. Like if I have a pathway and I want to show like what genes are actually changing and in which direction they are changing, I want one single picture for my presentation or for my paper. So suppose if I am actually interested in this pathway called as influenza pathway, and I want to see like if influenza pathway, what are the genes that are changing in the influenza pathway, which direction they are changing. So I can use a package called as path view. Okay, and so this part is actually same thing what we did, that we actually removed any NA, we removed any duplicate, we subset our actually gene list at a p-value cutoff of 0.05, and then we are running a path view on this. So basically this is the same thing that we were doing, and I will show.
The whole data?
No, so whole data you should take. Yeah, but what we are going to see is only one pathway now. Okay, we are.
Give the command to the pathways list.
Path view.
I will actually show you path view. So in this one, so we created this list here, and that list we are.
See anything.
Oh, sorry. So yeah. Okay. So in each pathway, you have a ID like this, and where you will get this ID from. So if you go back into your Keck pathway analysis here, and if you look at head, you will see like there is a pathway ID is there. So if you want to see any of this pathway ID, you can use. We'll use one of this. Okay, let's use that.
It could be go. It could be.
It's basically it's basically Keck pathway you have to use. Okay. So suppose I'm going to redo this analysis with this the one pathway which was enriched in our data set. So the only thing that you need to change, what happened? But anyway, let me show you. After that we'll come back. So it put this actually put your all the genes in that So what it did, yeah, I will just finish with the path view and then we'll come back. So what it did, it actually took your genes from the differential gene expression did and it laid it over the pathway. And what you are seeing here, that in this case, fortunately or unfortunately, all the genes are upregulated. But if there is a down-regulated gene, then you will get a green color there. So in one go, you can depict your whole enriched pathway or enriched pathway into single thing. And if you have 4 or 5 different pathways of your interest, plot each pathway individually. And how you do that, you will just change.
The ID.
Oh, where is it? Oh, where is this? So yeah, just change this pathway ID, okay, here. This pathway ID you change and you will get the new pathway. And there's one thing, this is the only package I know where you don't have to use that PDF or that the storing. It will directly store into your whatever working directory it is there, and it will show you that. So just change this. If it is a different organism, you change the species also. If it is mouse, then make it MMU. If it is Drosophila, make it DMA. Otherwise, human, keep as such like HSA. Okay. So we have done enrichment analysis using 3 different ways. We used GOTERM analysis. We did GSEA. We also did Keck pathway analysis. We learned 4 different ways of plotting them: dot plot, bar plots, EMAP plot, and CNET plot. And we learned how to interpret EMAP plot and CNET plot. And we also learned how to plot your Keck pathway data into path view. And then, so I think with this, if you can do this much, you are very comfortably, you can write a good paper also and use some of this. So these are like publication quality figures, so you can actually use directly for your publication or for. So I think we should go for a T, and we will actually come back and then Manpreet quickly show you that all these things what I told you, you don't be actually showing you this.
One more thing. We classify or we sell using